All right, hi guys. This video is going to be the first lecture of this unit. So we are starting a new unit on the cell cycle. We're going to look at overall the cell cycle, like big picture today. And then eventually we will have a day where we learn about meiosis and mitosis, which should sound familiar. You should have learned about at least mitosis in 7th or 8th grade. And we're actually going to kind of fly through that, and we'll continue on learning. But today we're going to focus on the cell cycle. Basically, we're going to look at like all of the cells in our body right now, what is happening with those cells. It differs depending on where the cells are found, how old the cells are, or lots of things. So let's look. So when we think of just overall cell growth, cells cannot just continue to grow and grow and grow and grow continuously. So as cells grow larger and larger and larger, it basically puts too many demands on the DNA. So what scientists have found is that it makes sense to have smaller cells, because those smaller cells then have less materials to move in and out. It makes it a lot easier to manage and to monitor and to run a tiny little cell than it does like one gigantic cell. So cells are tiny for efficiency, in order to allow the DNA to be most efficient and to allow stuff to move in and out of the cells more efficiently. So before the cell becomes too large, it'll undergo what we call cell division, and it's going to divide into two. Okay, so it'll be triggered for various things we'll talk about. For various things, that cell will all of a sudden undergo division and divide into two cells. So we'll start with one and end up with two. It will make an exact identical copy of the DNA, and each new cell gets one complete set of DNA. So if your skin cell, there's lots of your skin cells right now that are starting with one cell, dividing into two. The two new cells are genetically identical. They each have a complete set of DNA, okay? So cells must reproduce for various reasons. Number one, for repair, right? So every time that you're injured, your body is able to repair itself. Most of us, our bodies are really good at repairing themselves. So you bite your tongue, within a day, most of the time, a normal bite of the tongue has been repaired. General maintenance, so not even like if there isn't necessarily what we would call like an injury, just regular repair. Growth, so obviously your body is made up of millions more cells just because you're literally so much bigger than you were when you were the day you were born. And then again, formation of an adult. So just literally growing, increasing the size of your body by how many times. So some of these words, we're actually going to do a lot of vocab this unit, and actually specifically today. One of the vocab words, and I'm going to have you practice on a Quizlet, memorize, is chromosomes. And we've heard that term thrown around, but what that term actually is is a thread-like structure. Sorry. A thread-like structure within the nucleus that contains our genetic information, or the DNA, that's passed from one generation to the next. This is what we're going to call a chromosome. So the chromosome is like an X-shaped structure made up of our DNA. Each chromosome is composed of two identical chromatids. That's another vocab word you're going to memorize today. So chromosome is X-like. Chromatid is half of this. So this one side is a chromatid, and then this one side is a chromatid. the two chromatids are held together at the middle here by what's called a centromere. So this yellow thing here is a centromere. A small segment of DNA is called a gene and codes for a specific trait. So if you were to look at your chromosome, you could point to any location, and that would be like one gene, right? One location is coding for one gene. You're familiar with that. Oh, you get that gene from your mom, you get that gene from your dad, and that is true, right? So you get all of your DNA from both your parents. And one gene is just like one location on the chromosome that codes for just one specific trait. So chromosome, chromatid, another chromatid over here, held together by the centromere in the middle. Here's a picture on the screen. So the whole X-shaped structure is the chromosome. The chromatids, there's a left chromatid and then a right chromatid. They're held together by this green blob, which is shown here as the centromere. And then again, any point on that chromosome is what we call a gene. This kind of puts it in perspective. So basically we start with our cell. Inside the cell is the nucleus. Within that nucleus are all of our chromosomes. Within the chromosome is our DNA. And then one location on that DNA is what we call a gene. We'll look next unit at the structure of our DNA. Here's just another picture. So big X shape. And the picture on the right is from an actual like microscopic image of a chromosome. So you can see it actually is shaped like an X. Has a left chromatid and a right chromatid. It's given that it makes that X shape because it's held together here by the centromere. Okay, so chromosomes like this are only visible during mitosis, which is only like one tiny fraction of the time the DNA spends in the cycle. So like only very short period of time is our DNA in the shape of chromosomes and actually visible by like fancy microscopes. The pictures here actually on the screen, those are two more chromosomes. So we can take pictures of chromosomes just like this. Most of the time, our DNA is not in the shape of a chromosome, actually. I don't know why we're never taught this. It actually looks like this. So if you can kind of see my yarn chromosome, what I've done was wound up my yarn really, really tightly because that's essentially what happens to our DNA. But most of the time, during interphase, the DNA is in a disorganized mass like this. It actually like relaxes but becomes disorganized. And this is called chromatin. So most of the time our DNA actually looks like this and not what you normally think of as a chromosome. The reason for that is our DNA code is read constantly, 24/7, nonstop. Our code is read, and it has to be read like this. So, and we'll see this next unit. So you'll see that literally our DNA gets read one little, it's called a codon at a time. Okay. So when the DNA is in the form of chromatin, it's just functioning like DNA. Every type of organism has a specific number of chromosomes. Humans have 46, so we get 23 from mom and 23 from dad. So we have a total of 46 chromosomes. Every organism is different. It actually shows to be very random. Some things that are really like what we would think of as like simple organisms have like way more chromosomes, but so it doesn't, we don't actually see a significant trend about like the complexity of their brain or anything like that. Chromosomes are replicated before the division even starts, so that each cell has a copy. Okay, so chromosomes are replicated before cell division even starts. So here's an example of the number of organisms. Roundworms have 4, for example. Little rice has 24 chromosomes. Humans, 46. Chimpanzees, 48. Dogs, 78. Okay, so when we look at the cell cycle, we're looking at a series of specific events that our cells go through as they grow and divide. So all of our cells right now are at one phase, one of the phases of this cell cycle. During the cell cycle, cells are going to grow and function like a normal cell. Then they're going to prepare for division. And remember, in order to prepare for division, they're going to replicate the DNA. So like right now, a Miss Ryberg cheek cell wants to make a new cheek cell because we're constantly making cheek cells. So a Ryberg cheek cell right now is replicating the DNA, and it'll eventually have double the DNA crammed in there and eventually divide. And that way, the two new cells each have a complete set of Ryberg DNA. So those two daughter cells have complete sets, and then eventually the cycle will begin again. I mentioned our cheek cells constantly make new cells. That's one of the places in our bodies, like skin cell and almost all tissues along our like digestive tract constantly make new cells because of the acidic environment. So our skin cells are constantly producing new cells, constantly. Inside our mouths, tongue, the sides of our cheeks, and so on. Okay, so here's the cell cycle. Every cell in your body is in one of these 4 phases. You'll have to memorize this. So let's see. I like to start. So M stands for mitosis. So let's imagine mitosis is a cell making new cells. Okay, so we're starting with a brand new cell. Imagine we have 2 identical brand new cells. They're going to come out of mitosis and begin the G1 phase. If you look over here, G1 just stands for growth, and that's truly what's happening. So these are smaller cells for sure. They're going to, now if they're a Ryberg cheek cell, they're going to function like a cheek cell. Like they're able to function, but they're also just going to grow and like literally bulk up. They're going to reproduce organelles as needed, right? Eventually they'll become triggered to enter the S phase here. So lots of our cells right now are in the S phase. And the only thing that we identify as happening in the S phase is DNA synthesis. And think of synthesis as like producing or reproducing or making, okay? So, and that's what's happening. So here in the S phase, our cheeks, my Ryberg cheek cell is acting like a cheek cell, but also it is replicating the DNA. The only thing that happens in the S phase is DNA synthesis. Once the DNA has been replicated, appropriately replicated, there's lots of checkpoints along the way. There are lots of errors that happen, and usually our cells catch, and we'll talk about that, but usually our cells catch the errors and actually like kill the cell. Let's say everything is good to go. The cell gets a thumbs up that it's replicated the DNA properly. Then it's going to enter the G2 phase. The G2 phase again stands for growth. So it gets a little bit bulkier. Now it has like double the DNA. It's also going to continue to grow and produce more organelles and cytoplasm in there, get a bit bulkier in order to prepare for mitosis for when it divides. Then once it's good to go, it will enter the M phase, mitosis here. And that is when this one cell will divide into 2. Now there's a lot that happens in just one cell dividing into 2. So we'll spend a whole day just talking about a whole section of notes, day of notes, I should say, on just that one phase, just the M phase. And then the cycle starts over again. So the steps of the cell cycle, so G1, a new cell grows and develops. The S phase, the chromosomes are copied. The G1 phase, growth, preparing for division. And then the M phase is just mitosis. And again, in a couple days, we'll learn more specifics about mitosis. G1, S, and G2 together make up interphase. And so pretty much if it's not dividing, if it's not in mitosis, it's interphase. And we think of interphase as basically cells acting normal. If you look at, actually, let's look at the like the illustration to the left here. You can see inside the nucleus. If you look at the DNA is colored kind of, it's like shaded a little bit. That's representing DNA in the shape of this. So remember, this is called chromatin, and it's just like a disorganized mass, and you can't really see it in a microscope. That's why in these images, so here's a real microscopic image of cells. These are plant cells. You can kind of see the cell themselves is kind of like shaped like a brick, like square or rectangular. And then the darker circles, those are the nuclei. If you look at these two cells right here in the nuclei, you can't really see the chromosomes. Chromosomes will typically look like black wormy things, and it just looks overall shaded. So right now the chromatin will look kind of like dark and shaded. So again, with interphase, if you look at the inside arrow here, and I think this diagram is in your notes, and I think you need to click and drag G1, S, G2, and M. So I like this diagram because it shows interphase. So it shows that G1, S, and G2 collectively make up interphase. Then, if the cell is triggered, it begins to go through mitosis. So those cells are going to go through the phase and produce two identical daughter cells and then start the process over. The specific phases of mitosis might sound familiar. So we have prophase, metaphase, anaphase, telophase, and cytokinesis. These pictures here on the right are actual cells shown from a microscope. That's not actually that much stronger than what we would normally, if it weren't for COVID, what we would normally be using and like looking at in class. And what I see here, I'm going to just point out one thing, are the chromosomes. Remember I said chromosomes look kind of like dark, wormy looking things? The dark wormy structures here, like right here, those are the chromosomes. And same here, the dark structures, they look like dark worms. Those are the chromosomes. Okay, so to rehash some vocab and to add a few that I'm going to have you memorize by Thursday. So we're going to have a quiz in a couple days, a 6-question quiz, because you cannot move on in this unit if you don't know what these terms mean. So the term spindle fibers refers to a fan-like microtubule structure that goes from one end of the cell to the other during mitosis to help the chromosomes like organize and separate during anaphase. So I just need you to memorize that the spindle fibers are these microtubules that spread across the cell, and really they attach to the chromosomes, they move the chromosomes and divide them into two equal chunks. So eventually in mitosis, and you'll see this, but eventually in mitosis, we want like one clump of Ribberg DNA here and one identical clump of Ribberg DNA here. The spindle fibers are responsible for moving those around. In the pictures here, the spindle fibers are is like this line and this line. And you can see they stretch across the cell. Some of them are going to be attached. You can see in the left here, attached to the actual chromosomes and move them around. The second term is chromosome. So remember the chromosome, the key is that it's organized thread-like strands of DNA that form an X and they're visible during mitosis. These are the wormy looking things we saw in the images. Chromatin is now this. Okay, so that cell now enters interphase, starts functioning like a normal cell. And so the DNA kind of decondenses and relaxes into chromatin. So chromatin is the disorganized mass of DNA in the nucleus during interphase. A chromatid, remember, is just one individual strand of a chromosome, so one half. So here is to you, I guess, the left half and then the right half. So here's one chromatid. This is the second chromatid. The centromere is what holds the two chromatids together. And the centrioles. I will say over and over, they look like pills to me. So they're kind of like a pill-like looking structure. Here's an image of the centrioles on the left, but then in the right, here's the cell, and this is through mitosis. So what we have here are chromosomes in the middle. They're attached to the spindle fibers. And remember, those spindle fibers, that's one of your words. The spindle fibers attach to them and move them and organize them in mitosis. The spindle fibers come from the centrioles. So here are those pill-like looking centrioles. So the centrioles anchor the spindle fibers during mitosis. Here are the there's lots of C words, and this is what I'm saying. It wasn't until last year, I think, that I had the strike of genius to make you guys memorize these terms now, like literally today. You will be so confused for the next week or two if you can't get these terms straight. So I want you to memorize chromosomes, chromatin, chromatid, centromere, centrioles. There's 5, and I call those the C words. But then for the quiz in a couple days, I actually added spindle fibers. So I also added 1 S word. So there's 5 C words and spindle fibers. There's a Quizlet posted that should be posted with just those 6 terms. You gotta memorize them. And then we'll have a quiz during class on Thursday on literally those 6 terms. And that's it. All right, guys. So that was the introduction to the cell cycle. Again, after this, we'll focus for a few days on mitosis, on just specifically mitosis, and then meiosis, and so on. So, all right, guys, thank you. Let me know if you guys need anything.